We're now going to shift topics a bit from talking about the cell cycle to talking about something slightly different, which is genetic diversity. And so in this part of the course, the thing that we're going to be concentrating on, the question we're going to be concentrating on, is how can we account for changes in organisms' genetic makeup through time? And so I want to start this topic with some puzzling observations. So the first comes from karyotype examination. So karyotypes are basically taking cells that are on the verge of dividing. So after the chromatin has condensed down to chromosomes, and then there are a bunch of different substances that you can use to stain chromosomes that will label the DNA that's there. So you can stain all the chromosomes. And when you do that, each chromosome winds up having a very distinct banding pattern that will form on the chromosome. And so if you stain a bunch of chromosomes, then and then you can, when they're in a cell, they'll often be all over the place. So you can then arrange them, cut them. People would actually cut out pictures of the chromosomes and arrange them. And what they found is that if you look at the karyotypes for a bunch of different kinds of cells, you'll find that some cells have pairs of chromosomes that are the same size, while other cells only have half as many chromosomes, and each chromosome is a unique size and shape. And then along with that, another thing that biologists have noticed for a long time is that for some of the multicellular organisms that we interact with, the parents look different from their offspring. So, we even see this in ourselves. Human offspring might resemble their parents, but they look different. So, just to give you an example of what I'm talking about with karyotype examination, earlier when I was talking about chromosomes in eukaryotic organisms, I mentioned the example of this ant species, the jackjumper ant. And I mentioned that females have 2 chromosomes, and their 2 chromosomes are the same size, and then males only have 1. And that's actually true across ant species, and it's also true across bee species as well, and across wasps. So in all 3 groups, the females have paired chromosomes, while the males just have individually unique chromosomes. They have different total numbers of chromosomes, but that general pattern is consistent. This brings us to a point where we need to talk about a little bit more chromosome terminology. So I've got some of these terms here. So homologous chromosomes. So as I say here, they have the same size and banding pattern. So I've got a picture here of human chromosomes, someone who is kind enough to post their own karyotype on Flickr, and these are all arranged into pairs, and so hopefully you can see that the banding patterns on these pairs are very similar to each other. Another thing you might notice here is in some cases the centromere is in the middle of the chromosome, but in other cases it's actually closer to one end, and that's very common to have happen. So there's no rule that the centromere has to be in the center of the chromosome. It can be anywhere along the length of the chromosome. So that's homologous chromosomes, so same size and banding pattern. But then there's something else that happens, and that is what I refer to here in terms of ploidy. So ploidy refers to the number of sets of homologous chromosomes. So here's how this naming convention works, right? So, and if an organism is haploid, it stands for half. All their chromosomes are unique. There aren't any pairs. So this is often symbolized just using the letter N. Now, if they are diploid, so di means 2. So if they're diploid, they have pairs of homologous chromosomes. So we are diploid. We have all of our chromosomes come in pairs. And so this is symbolized 2N. And it can actually get more wild than that. So There are organisms that are triploid. So their chromosomes come in sets of 3. So that would be symbolized 3n. And we can keep going. So you could be tetraploid. So chromosomes occurring in sets of 4. And then kind of past that point, we don't come up with specific names anymore. So then we just call everything polyploid. And so poly means many. So multiple homologous chromosomes, more than 4 per set, right? So if we doubled all of these human chromosomes here, then we would have a tetroid human. Okay, now I want to actually put a little caveat on what I said a minute ago about parents looking different from their offspring, because this isn't actually always true. We can divide types of reproduction into 2 major forms, depending on the organism and the context. So many organisms reproduce using asexual methods. So A means without. And organisms that reproduce asexually have offspring that are identical to the parents. So here's one of my favorite examples, this plant here. So this is an agave plant. You might know these for being used to make tequila. And agave can reproduce by growing baby plants around the base of the parent plant. So these baby plants are actually called pups. And these baby plants, they're identical to the parent plant. And so that's one method for reproducing. Agave can also reproduce sexually, so they're kind of strategic. They've got multiple methods to produce offspring. But then for organisms that reproduce sexually, a big part of what sexual reproduction accomplishes is that it allows for new combinations of traits in offspring that are a mixture from 2 parents. And I actually want to talk about 2 other unusual examples of asexual reproduction. We often think of that as being confined to plants, but it's really not. So here's a news story from a couple years ago about the discovery of an asexual species of termites. And I just thought this headline was really funny. And here is actually an example of a lizard species that reproduces asexually. So this center lizard here is female, and she'll just lay eggs that are new females that are identical to her. The 2 species on the outside are species that normally reproduce sexually. Okay, so we've got these 2 puzzling observations about pairs of chromosomes in most cells, but then others that only have half as many chromosomes. So what I'm saying here is, in the majority of cases, organisms are diploid, but there are some cases where we can find cells that are haploid. Okay? All right, so why does this happen? And when I'm asking this causal question of why this happens, we're going to mostly focus on the proximate question here. So, how is it that cells go from being diploid to haploid and back again? And the thing is, we can't explain these observations with mitosis, right? Because with mitosis, we go from a single parent cell to 2 identical daughter cells, right? So there's no change in the total number of chromosomes and in chromosome pairing through mitosis. Every daughter cell gets a copy of every chromosome. So what that leads us to is the idea that there must be a second set of things happening to allow for new genetic combinations to arise. And those 2 things are the processes of meiosis and fertilization. So here's the thing about meiosis and fertilization. So these are processes that alternate, and so organisms will go between a diploid state, where they have chromosomes in pairs. Then they'll go through the process of meiosis, and then they'll be in a haploid phase of their life, So at that point, their chromosome number will be designated N, and then fertilization will happen, and through fertilization, they'll go back to being diploid. But the amount of time that an organism spends as a diploid or as a haploid can vary. So we're going to start out thinking about humans, because I think it's a little easiest to start out thinking about ourselves, and then we're going to look at some other methods for managing this process of toggling back and forth between being diploid and being haploid. So we spend most of our lives as diploids. So our cells, our bodies are mostly made out of diploid somatic cells. So a somatic cell. So the word soma actually means body. So somatic cells are most of the cells that make up our body. But there's a contrast. So we have a group of cells in our body that are referred to as germ cells. And those germ cells then undergo meiosis and form haploid gamete cells. So oops, sorry, they're not 2N, they are N, okay? So, the gametic cells in humans, we typically think of as being eggs and sperm, but kind of the collective term for eggs and sperm are gamete cells, and they're haploid. So then, so most of our body, it consists of these somatic cells. We've got a subset that are haploid gametic cells. So when an egg meets a sperm during the process of fertilization, then the result of that's going to be a new diploid zygote. So a zygote is a young developing offspring that's then diploid again. And that zygote will grow up, they'll mature, and then the whole cycle will repeat. Okay, but here's something a little strange and different. So this is the life cycle for plants and for some type of algae. These organisms divide up their generations equally. So both the diploid stage and the haploid stage have multicellular forms. So we wouldn't really talk so much about this contrast between somatic cells and germ cells, because that concept's not really relevant for these plants and algae. And so here's an example of that. So what's in this picture right here are all a type of fern, and you can see there's some large fronds here, and then there's something really tiny down here that's also growing, that's multicellular, that's also a fern plant. And this fern plant is the haploid form of the larger fern that you see pictured here. And so this multicellular haploid organism up here is referred to as the gametophyte. You don't need to worry too much about the term gametophyte for this course, but I think it's just useful for understanding this life cycle type. And then in contrast, the larger forms here that will eventually produce spores, so they'll go through meiosis and then they'll produce haploid spores. Those multicellular organisms are referred to as the sporophytes, right? So they'll release spores. The spores will travel some distance, get established, and they will grow, and they will be multicellular gametophyte. And at some point, two of those gametophytes will interact with each other. There will be a fertilization event, and they'll go back. That plant will go back to a diploid generation. Okay, so you don't have to be unicellular as a haploid. And then there are other organisms where the haploid form can predominate. So this again goes back to ants and bees, as I was saying about the haploid form. So the male ants and male bees, they spend their whole lives as haploids. So they develop, they mature, and their entire life they are haploid. And they actually form from unfertilized eggs from a queen ant or a queen bee. So they just completely have skipped the fertilization step. They do fertilize ants, the queen ant and queen bee, to produce diploid females, but the males are haploid. And the same is also true, as I've illustrated here, for some fungi and for protists as well. So in those forms, most of the lifetime is spent as a haploid and just a little bit of time as a diploid. Okay, so that's kind of an overview for thinking about meiosis, and now we're going to dive in a bit further to explore how meiosis compares to mitosis. So here's a little diagram for this. So on the left I've got mitosis as a bit of review. So for mitosis there's just one round of prophase, metaphase, anaphase, and telophase. Hopefully you're envisioning everything that happens during those phases as I say those words. At the end we wind up with two identical daughter cells, and there are two new cells at the end of mitosis. So in contrast, in meiosis, cells go through two rounds of prophase, metaphase, anaphase, telophase. And by the end of meiosis 2, the end result will be unique haploid daughter cells. And we'll take a look at how that happens. And so if a cell goes through two rounds of PMAT, then by the end of round 2, the end result will be 4 new cells. And so those 4 new cells are potential gamete cells, okay? And that whole process has to be followed by fertilization at some later time. The exact timing will depend on the organism. Okay, so we've got meiosis 1 and meiosis 2. And here's kind of my summary for the major events that take place in meiosis 1 versus meiosis 2. And we're going to be looking at these in much more detail in a moment. But this is kind of a higher level summary of what happens. So in meiosis 1, the two big events that take place are a process called synapsis, also known as crossing over, where homologous chromosomes will pair and they can actually swap DNA. And we'll look at that more closely. And then the other thing that happens during meiosis 1 is that the homologous chromosomes separate from each other. And so because those homologous chromosomes are separating from each other, this is the point where cells go from being diploid to haploid. So meiosis 1 is often referred to as the reduction division because this is taking place. And this is kind of a side note, but in some organisms there can be a pause between meiosis 1 and meiosis 2. It really just depends on that specific organism. Okay, so that's meiosis 1. Meiosis 2 is a little bit simpler. It's very similar to mitosis in that the main thing that happens in meiosis 2 is the separation of sister chromatids. So what that means is that in meiosis 2, chromosomes will go from containing 2 pieces of DNA to containing just 1 piece. And I've got a link here on this slide just in case you want to see this process in action. It can be really helpful to practice. I still recommend gummy worms, but a video or 2 can't hurt. Okay, so let's dive into a draw along for meiosis 1 and meiosis 2. And we're going to work with a diploid cell, so 2N, that has 4 chromosomes total, so 2 pairs of homologous chromosomes. And we're going to follow 2 physical locations on one set of these chromosomes. So I'm going to put this draw along in its own video so that you can come back to it when you're ready for it.